Hi. As you can see in this video, we'll talk about cell function and eukaryotic evolution. This is part of the cell unit that when we're talking about cell membranes, we spend so much time talking about the structure and diffusion and transport that we didn't really get to talk about the different cell organelles. And so this kind of gets put here because a lot of the organelles have membranes as part of them, and that's where the connection is. So we'll start off with the idea that there are two basic types of cells. You have prokaryotes and eukaryotes. Prokaryotes being the first types of cells, bacteria and such, and then eukaryotes being animals and plants and fungi and things like that. So the prokaryotes were the first cells to evolve, and they are the simplest. They have basically the minimum that you need to be alive, which means the minimum you need to make proteins and protect yourself. You have your cell membrane, and then you have DNA and ribosomes, right? That's all you need to make proteins, or DNA and ribosomes. The cell wall's there for protection. You might need to move around. The flagella might be on the bacteria, might not. But really all you need are the DNA and the ribosomes and the cell membranes, right? But doing just the minimum is not usually the best strategy. And so eukaryotes come along and have much more than the bare minimum. Just like before, you have your cell membrane and you have your DNA and your ribosomes. But now you have all these extra things. The most famous of which, of course, is the nucleus that surrounds the DNA. Then you have the endoplasmic reticulum and Golgi and these things right here, right, which are all extra, all things that prokaryotes don't have, and they're all things that require membranes to make them compartmentalize, right? And so one of the big defining characteristics of eukaryotes are membrane-bound organelles. And so we're going to talk about each of those. Most of them have to do with making proteins. Of course, you have your nucleus and your ribosomes, your DNA and the ribosomes to build the proteins. But then we need all these other structures to help us to make proteins even more efficiently. The main structure here would be the endoplasmic reticulum. This is actually just an offshoot of the nucleus. It's all one big part of the membrane. That's all just folded to make it look like the nucleus is a big circle with the rough endoplasmic reticulum around it. But it's really all just one continuous membrane that's just been folded in a very complicated way. It's called the rough ER because it has ribosomes attached, which means that this main job here is to make proteins. The messenger RNA coming out of the nucleus would immediately go into the rough ER and find a ribosome, and the ribosome would start to translate that into a protein, right? There is another part called the smooth ER. This one is not as commonly brought up because it's actually really complicated. You just need to know two things, which would be lipids and detoxification. The main one being the lipids, because that's where we make membranes, right? Because we got to make some phospholipids. So this is where membrane components are made in the smooth ER. But again, most of these are for proteins. So once it leaves the rough ER, then it's going to need to go where it needs to go. And that's the job of the Golgi. Again, you kind of talk about it labeling and shipping things. Again, you don't need to know how that happens, but the idea is that the protein gets brought to the Golgi and the Golgi sends it off where it needs to go. The Golgi is an interesting looking organelle. Again, it really highlights the idea of having a lot of surface area, because rather than having one just big glob, it's a bunch of little pieces that are all stretched out to give it as much surface area as possible. The Golgi relies heavily on vesicles, and vesicles are basically just small bubbles of membrane, right? Like little phospholipid bubbles that float around. In this case, it's bringing in a food particle that needs to be digested, or over here, it's sending the protein from the rough ER to the Golgi, so that it can then be packaged off by the Golgi to go wherever it needs to go, right? So transporting things inside the cell is done by these things called vesicles. A lysosome is a special type of vesicle that is filled with digestive enzymes. This is the waste removal and recycling center of all the cells, right? And so just like before, we have the food particle that's being transported in from a vesicle, but then that vesicle fuses with this one. And then that is trying to show that the big food particle gets broken down into its monomers, right? It's being digested into its basic components. You might want to break down a whole organelle, right? Like if the cell, something breaks, you can just completely recycle just anything inside of a lysosome. And so, and again, we have our eukaryotic cells that have all this extra stuff going on compared to the prokaryotes. Most of those, again, are for protein efficiency, protein synthesis efficiency. But then there are a couple organelles that I didn't mention yet that are super important and that definitely characterize a eukaryote from a prokaryote. And that would be the mitochondria and the chloroplasts. Right, so this part then starts to talk about how did we get to be the modern eukaryotes? How did we go from the basic prokaryote to the really complicated eukaryote? Step one would be to start to get the membranes that makes compartments, compartmentalization, right? And so I like this graphic. It makes it really simple that you have your ancient prokaryotic cell back then that just has DNA and ribosomes inside of the cell membrane, but the membrane, the ribosomes are still stuck to the membrane, right? I like this idea that the DNA isn't just floating around in there. It's got to be attached to something. And so then as the cell membrane starts to fold and become more complicated, you get more and more compartments that are made. And again, I really like this picture because it shows that the endoplasmic reticulum is just an offshoot of the same membrane that you would make the nucleus out of, right? Making compartments leads to smaller areas with better surface area, which leads to more efficiency, right? So that's the main idea of eukaryote, is that we have all these membrane-bound organelles. Now, again, the two really important organelles are the mitochondria and the chloroplasts. And these are interesting because there's something called the endosymbiotic theory, that the first mitochondria started as an aerobic bacteria that was out there just doing its thing. And then it got swallowed by a big primitive ancestral eukaryote. And when the big eukaryote swallows the bacteria, instead of eating it, it just kind of holds it inside there. And the bacteria, which is really good at making ATP using oxygen, just stays in there and starts to do that. Whereas the eukaryotic cell gives the bacteria a home and provides it with food and things like that. Right, and something that you could see in the evolution of that is that there's the mitochondria have the two, the outer and the inner membranes. The outer membrane was the membrane that was left over from outside here, and then the inner membrane is the real membrane of the bacteria that now has this extra part on the outside. At some point then, a cell that had already done that with the mitochondria again swallows up, this time a photosynthetic bacterium, and that bacteria ends up, instead of being digested, forming a relationship where it produces all the sugars via photosynthesis that now that cell can benefit from. And then of course the bacteria benefits by getting their protection and food sources. Again, this idea is shown in the complicated membrane structures of the mitochondria. In both the mitochondria and in the chloroplasts, the inner membrane, the original bacterial membrane, is where you'll find the electron transport chain, right? Both places, it's on the inner membrane, right? The outer membrane is just where the original eukaryote way back then swallowed them, right? So that membrane doesn't do a whole lot. All the magic is happening on the inner membranes. And again, these membranes show the idea that having a lot of surface area is important, because since this is where you'll find all those electron transport chain proteins, the more surface area of the inner membrane, the more proteins you can have, and the more electron transport chains you can get, and the more ATP you can make. Again, same thing here, where the thylakoids are basically just really, really folded up membranes. And the more you fold them, the more surface area they get, and the more electron transport you can do and generate ATP, which of course in this case is going to be used to make the sugars. But again, membranes are a huge part of cells. Surface area is a huge part of membranes because that's where you find all the proteins. And of course, all these membrane-bound organelles are there mostly to make proteins as efficiently as possible. And so there you have it. Cell organelles and the evolution of eukaryotes for you.